Good morning, viewer. Welcome to our Globe TV. Here's a breaking news. Yes, in a study conducted by MIT's Pickover Institute for Learning and Memory, researchers found that a protein named perlecan plays a crucial role in maintaining the structural integrity of neurons. Perlecan is part of the extracellular matrix that surrounds cells and helps them develop in a supportive yet non-rigid environment. The study revealed that without perlecan, the axons, long projections of neurons used for connection, can break apart during development, leading to the death of synapses, neuronal connections. Perhaps the most obvious feature of a neuron is the long branch called an axon and ventures far from the cell body to connect with other neurons or muscles. If that long, this projection ever seems like it could be vulnerable. A new MIT study shows that its structural integrity may indeed require the support of surrounding a protein called perlecan. Without that protein, in Drosophila fruit flies, researchers at the Pickover Institute for Learning and Memory found axonal segments can break apart during development and the connections or synapses that they form end by dying away. Perlecan can help make the extracellular matrix, proteins and other molecules that surround cells stable and flexible so that cells can develop and function in an environment that is supportive without being rigid. What we found was that the extracellular matrix around nerves was being altered and essentially causing the nerves to break completely. Broken nerves eventually led to the synapses retracting, says study senior author Troy Littleton, the Meningcon professor in the MIT's Department of Biology and Brain and Cognitive Science. Humans need at least some perlecan to survive after birth. Mutations that reduce but don't eliminate perlecan can cause Suzuki-Jampel syndrome, in which patients experience neuromuscular problems and skeletal abnormalities. The new study may help explain how neurons are affected in the condition, Littleton says, and also deepen scientists' understanding of how the extracellular matrix support axon and neural circuit development. Eliane Ghosh, PhD 23, who recently defended her doctoral thesis on the work, led the research published on the June 8 in the journal Alive. At first, she and Littleton did not expect the study to yield a new discovery about the durability of developing axons. Instead, they were investigating a hypothesis that perlecan might help organize some of the protein components in synapses that fly nerves develop to connect with muscle. But when they knocked out the gene called troll that encodes perlecan in flies, they saw that the neurons appear to retract many synapses at a late stage of larval development. Proteins on the muscle side of the synaptic connection remain, but the neuron side of the connection withdraws away. That suggested that perlecan had a bigger role than they first thought. Indeed, the authors found that the perlecan was not particularly enriched around synapses, where it was pronounced was in a structure called the neural lamella, which surrounds axons bundles and acts a bit like a rubbery cladding around a TV cable to keep the structure intact. That suggested that a lack of perlecan might not be a problem at the synapses, but instead causes trouble along axons due to its absence in the extracellular matrix surrounding nerve bundles. Ben Dasiport, Globe TV, Delhi, India.